Picking up where we left off on video 1, let's start talking about changes in muscle acid balance, and really we'll start getting into mechanisms of change, what's actually driving some of these changes that we're seeing with endurance training. If you go back to last week when we were talking about acid-base balance, realistically, our pH is going to be maintained in a very, very, very tight window. 7.4 plus or minus like 0.05, right? Like there's not a huge deviation that's going to be occurring in acid and base balance. Or we're not going to get super acidic. We're not going to get super basic. I mean, some of us might be because it is pumpkin spice season. I used that joke twice in 2 weeks. It's still good, right? Realistically, we're going to have these buffering systems in place. Like we know that we have a high level of bicarbonate buffering available. We have these transporters, we have these antiporters and these symporters that are all going to be capable of handling the acid load and really helping force us towards this more alkaline environment, or the 7.4. Because remember, that's still slightly alkaline. 7 would be perfectly in the middle for acid and base because it's 1 to 14. But if we think about where's some of this acid coming from? We know that it's coming from CO2 production, which regardless, when we're doing endurance training, we're not going to be able to shut off CO2 production because we have to make it, because we have to make high levels of NADH, because we have to make high levels of FADH, and we have to use the electron transport chain. We're going to make CO2. We're not going to change that. We're not going to change that ability at all. We're not going to change how much muscle contraction is occurring because that ATPase is making ADP plus a hydrogen, right? But what we are going to be manipulating is that pyruvate to lactate formation, right? If we're increasing the number of mitochondria, we're increasing our ability to burn fat, and we're making citrate, we're producing citrate from that fat burning, we're actually going to be inhibiting glycolysis from occurring. We're going to be inhibiting carbohydrate oxidation and carbohydrate burning from occurring. Because remember, glucose or glycogen has to get broken down into pyruvate and then get sent over to become acetyl-CoA. Fat can get cleaved into these two carbon segments and straight into acetyl-CoA. So if we inhibit glycolysis from occurring, we inhibit pyruvate to lactate formation. If we inhibit phosphofructokinase, we inhibit glycolysis from moving forward. So we're going to be able to essentially minimize the level of acid. We're going to be able to reduce the level of acid that we're actually would be producing. Didn't really rhyme, but it was like reduce and produce. I don't know. Something else that's really kind of cool here is that let's assume that fat burning didn't produce citrate, right? Let's just assume that fat burning didn't produce citrate. And we increased the number of mitochondria, like we have mitochondrial biogenesis with endurance training. What is that mitochondria really doing with the electron transport chain? Well, it's pulling away NADH, and it's using NADH, right? So we're going to be putting NADH into the electron transport chain so that we can generate high levels of ATP. If you remember, I made you guys focus on the ratio of NADH to NAD. And if the ratio of NADH to NAD got altered and there was too much NADH, we would actually increase the production of lactate. So by maintaining that NADH to NAD balance, by being able to use more NADH because we have more mitochondria, we're actually able to reduce the conversion of pyruvate to lactate even further, right? So we're actually minimizing this. And that would make sense because lactate is not a dead-end molecule, right? It's going to get recycled back to the liver for the Cori cycle and produce glucose, but we're already sparing glucose. So we don't need to have increased blood sugar, but we need to have blood sugar that's available. So it all circles back, right? Like this is where it all starts circling. So now let's think the molecular basis of adaptation. Why do these changes occur? Not just that they're occurring, but here we're going to take a deeper dive into why they're occurring or how they're occurring. In general, the major picture here is stress or disruption of homeostasis stimulates cell signaling pathways, right? So essentially, our body gets put into a state of stress, or we put our body out of homeostasis intentionally with endurance training or resistance training, it doesn't matter. We are intentionally stressing our system. That stress level actually induces a change. It causes cell signaling pathways to happen. So it's kind of like our cells tell our DNA, we need to adapt, we need to overcome, we need to evolve. We need to make ourselves better able to handle this stress. And the way our body does that is it uses something called a transcription factor, which is able to go in and manipulate the DNA and express different parts of DNA. And then when we change the expression of DNA, what we're doing is we're changing the proteins that are being made, because DNA is going to make proteins. And those proteins actually form the response to the stressor. So in general, the major molecular basis here is that we're putting ourselves into a state of stress. Our body is signaling to our DNA, hey, we need a change. Our DNA makes new protein, and we respond to that stress. The problem is that this is not rapid. This is not going to accumulate after one session. Change is not going to happen after one session. But if we start stringing together multiple training sessions, that stress starts becoming bigger and bigger and bigger, and our body has to adapt. It gives our body time to adapt, right? So the changes that you see with endurance training, it's not like you can go out and run 1 mile and the next day you're running 100. It takes a series of adaptations. It takes a series of stressors in order to produce these large sweeping effects. This is where the specificity of training comes in, right? If we're training everything, then you're not going to get significantly better at everything, right? You need to train single modality or minimal modality in order to get better at something. Even CrossFit, right? I love CrossFit, but you have to go through periods of time where you specialize in different movements and in different realms and different energy systems. If you look at all the top level folks, they're doing this. Essentially, if you want to get better at resistance training, you resistance train, because you have to give your body time to adapt molecularly. If you want to get better at endurance training, you have to do endurance training, and you have to continually stress your system in that manner to sum together all of the adaptations that you need to get better. How are we going to send this signal? How are we going to change the signaling that's going on? Well, we have really 4 major signals for adaptation. We're going to have mechanical stimuli, calcium, which you guys should automatically be like, I remember this, free radicals, and then phosphates and muscle energy. So mechanical stimuli, like this dude over here on the right is clearly struggling with a squat. He's clearly putting a high level of mechanical stress onto his muscle. It is that exact mechanical stress that is needed in order to signal to our DNA that we need to increase the amount of muscle protein there. That's going to be what is actually increasing muscle growth or anabolism. The same thing if it's increasing the stress with, it's the same thing, increasing stress on the muscle mechanically from squats or repeated landing of feet, like repeated footfalls, right? Or repeated contractions that are occurring as you're on a bike, or repeated contractions that are occurring as you're rowing. All those things, we're exerting a level of force, a varying level of force that we're going to have to adapt to. If we don't adapt, we die. Calcium, everyone should remember where calcium is coming from and how we're using it and how excitation-contraction coupling is playing a huge role here. Just because you remembered it for the first test or the second test doesn't mean it's going away for the third test, right? It's always going to be present. Essentially, what we have is that the level, the amount, and the duration of contractions that are occurring is going to vary the level, the amount, and the concentration of calcium. When that endurance training bout is going on and on and on and on and on, you're going to have high levels of calcium that stay present. And that's exactly what you're looking for here, right? So as a signal, free calcium is going to actually increase protein synthesis, which is going to help repair damaged tissue. Protein synthesis means repair. It means growth. It means bettering. It means making our body stronger. Free radicals. So free radicals are essentially unpaired electrons. We're going to create these as we're burning high levels of, or as we're making high levels of ATP, and as we're stressing our body, we're actually going to be creating these unpaired electrons. And realistically, what these represent is a deviation away from homeostasis, and they actually represent a bad state. So when you have high levels of free radicals, it actually is damaging to the body. But there's a concept that's called hormesis, or the hormetic effect, which is essentially that wonderful Kanye or Kelly Clarkson song that what doesn't kill you makes you stronger. It's the same thing here, right? You have to have a level of toxicity. You have to have a level of free radicals present in order to adapt. So it's the thing about like antioxidants, right? Or even better, ibuprofen. If you habitually take ibuprofen before exercise, after exercise, to handle pain associated with exercise, you're never going to adapt to the same degree as if you didn't take that ibuprofen. It actually blocks some of the negative signals that we need to have in order to get better. So free radicals, another example of how we essentially are able to signal to our body to adapt, right? Because they're bad, we need to get rid of them better. Phosphate and muscle energy levels. Remember AMP, ATP, ADP, ATP ratios, all this. Essentially, muscle exercise, muscle activity is going to accelerate the depletion of ATP. So we're going to get increasing ADP levels, or we're going to get decreased ATP to ADP ratios. And we're also going to get increases in AMP. And AMP is adenosine monophosphate. And what those represent is low energy status. Every single one of you guys that answered this question on the essay told me it represents low energy status, which is awesome. Now, the next level is that low energy status, that low energy level, is actually going to stimulate something that's called AMPK, or AMP kinase, or adenosine monophosphate kinase. And what AMP kinase is it is the energy sensor within our body. And what it does is it alerts our body to our overall energy status. So if we're low energy, we're going to signal, oh my God, we need more energy. And we're going to actually start producing more energy. We're going to start burning more fat. We're going to start burning more carbs. We're going to start burning everything. And that's exactly what these phosphate and muscle energy levels are going to do. We get better at handling this stress. As we get better at endurance training, our body better adapts to handling these low energy states. So now let's, that was supposed to be a dive. Let's go a little bit further, right? How is it that we're actually signaling? Well, we're going to be using second messenger systems like we talked about already in the hormone and the cell signaling section, right? This could be G protein coupled. This could be cyclic AMP, this calmodulin. All of these are going to start using some of that second messenger system signaling. So realistically, there's 7 of these second messenger systems that are going to simulate or stimulate is a better word, the muscle to grow and adapt and become better. And that's going to be AMP kinase, mitogen activated protein kinase 38, or MAP38 or P38. If you Google P38, you'll get a really cool fighter plane, I guess, because it's not a jet. It's worth a Google. PGC1 alpha, calmodulin dependent kinase, calcineurin, NF kappa B, and mTOR. So AMP kinase, again, central energy sensor. It's super important for our body. It's going to regulate basically every energy pathway, whether it's producing or consuming. It's going to stimulate glucose uptake. We're going to pull that glucose that's inside of the blood into the cell because we're in a low energy state. We're trying to get away from the tiger, but we don't have the energy. We're going to stimulate the breakdown of fatty acid oxidation, or stimulate the breakdown of fatty acids. We're going to actually start burning these fats. Again, we got to get away from the tiger. We don't have any energy. And it's also going to increase gene transcription to produce energy. Makes sense. And so what I want you to see over here is this is called a mechanism, or this is really a pathway map. And what you're doing is you're looking at what is going on inside of this biochemical pathway? How is this one molecule affecting other molecules? Anytime you see an arrow, it means that it stimulates it or it increases it. Anytime you see this bar right here, it means that you're inhibiting it. So AMP kinase is going to activate or increase PGC1 alpha, which is going to be responsible for mitochondrial biogenesis. It's going to increase your glucose transporters. It's going to increase glycolysis. It's going to increase fatty acid uptake. It's going to increase fatty acid oxidation, all of that. So it's going to do a lot to regulate our energy. P38, or mitogen activated kinase P38, is going to use the cyclic AMP second messenger system, which, if you go back, essentially you have cyclic AMP, activates protein kinase A, and then you go through a series of things, and you actually activate P38 MAPK. And what that is going to do is it's going to go into the nucleus. This is going to be the nucleus right here, and it's going to increase the transcription factor, the gene editor, PGC1 alpha. PGC1 alpha, look at how I did that, right? PGC1 alpha is going to be a key molecule, a key regulator when it comes to both high and low intensity exercise. Essentially, what AMP kinase is to energy sensing, which is the holy grail, PGC1 alpha is the equivalent to mitochondria. Essentially, PGC1 alpha is going to be the master regulator of mitochondrial biogenesis. If you increase PGC1 alpha, you increase the production of mitochondria. We increase the ability to burn fuels. We get better at burning, right? It's like souping up our engine. It's exactly what you're going to be doing when you increase PGC1 alpha. PGC1 alpha is also going to be responsible for signaling to form new capillaries, transition towards type 1 fiber types, and actually start handling antioxidants a little bit better. So when it comes to endurance activity, we want to maximize PGC1 alpha. And you can see changes in NADH ratios are going to play a key role in PGC1 alpha. Calcium is going to play a key role in PGC1 alpha. AMP ATP ratios, energy status, insulin, oxidative stress, or those free radicals, and then mitochondrial DNA synthesis, mtDNA, mitochondrial DNA synthesis, is going to be what occurs. This is going to be the downstream. Calmodulin-dependent kinase. What calmodulin-dependent kinase is going to do is it's going to essentially respond to the amount of calcium that is present inside the muscle cell. So as we increase the amount of calcium, we're going to increase the activation of calmodulin-dependent kinase, and that's going to increase PGC1 alpha. This is actually kind of a cool figure because it summarizes really everything. So exercise increases calcium, which increases calcium-dependent kinase, or calmodulin-dependent kinase, which is going to increase PGC1 alpha, which is going to increase nuclear genes for mitochondria, which is going to create new mitochondria, which means we're going to get an increase in respiration, which means that we're going to be able to actually increase the amount of peroxide or the amount of free radicals, increase the amount of peroxide, free radicals get handled by MAP, right? Like you can see how this kind of creates a circle. It's really important that you understand, at least at the basic level, that this signaling for calmodulin-dependent kinase and for PGC1 alpha and P38 is all going through PGC1 alpha to create more mitochondria. And then once you understand what mitochondria can do, you can start understanding how this benefits endurance activity. Calcineurin is going to be responsible for removing phosphates from a molecule. What I want you to know is that it's used to actually help restore and regenerate muscle. And it's going to be one of the keys in the transition from fast to slow fiber types. So what you can see is this is a tonically active. So this is a constitutively active, or this is a chronically active motor neuron. It's a better way to understand that. When you have that increased activation of a motor neuron, you're going to increase the amount of calcium, right? We're using excitation contraction coupling to increase calcium release, to increase binding to troponin, to increase conformational shift in tropomyosin, to allow actin-myosin binds to happen, to to use ATP to create contraction. You already know that. All that this is saying is that you actually use that calcium. Some of that calcium activates calcineurin, which activates specific genes inside of the body or inside the nucleus or inside the DNA to enhance slower oxidative muscle genes. So we actually facilitate the transition towards slow twitch fibers or type 1 fibers. NF-kappa B is what PGC1 alpha is and what AMP kinase is, but it's like essentially like that holiness is what NF-kappa B is to inflammation. So it is kind of a massive transcriptional regulator or a massive DNA regulator when it comes to inflammation, when it comes to oxidative stress, when it comes to viral challenges. This is going to be the mecca, right? So as you increase activation of NF-kappa B, as you increase the amount of NF-kappa B movement to the DNA, you're actually going to be increasing your defense. So you're going to be using your immune system to respond to this. You're going to increase anti-inflammatory responses, antioxidant defenses, all of that. So the level of inflammation that you're actually able to create within your body as you're doing training, the amount of stress that you're able to produce in your body is going to increase NF-kappa B. And then you're going to get the actual anti-inflammatory properties of exercise. So acutely, exercise is extremely inflammatory, right? It's an extremely negative stress on our body. It actually hurts our body to exercise. But NF-kappa B kind of helps take and turn the tables and make it anti-inflammatory in the long run. This is why they say exercise can be anti-inflammatory. And then mTOR. So this is our major protein kinase. And this is going to come into play a lot more in endurance training or in resistance training than it is in endurance training, but it still plays a role here. Essentially, it is the master regulator of protein synthesis. So if you want to get big, if you want hypertrophy, you have to stimulate mTOR. If you don't stimulate mTOR, you're not going to get that same muscle growth. So it can be activated a ton of different ways. It can be activated with diet, exercise training, as well as mechanoreceptors. So that muscle stress that we talked about, that mechanical stimuli is going to help activate mTOR. And what's funny is that it's actually inhibited by AMP kinase. So our body has to be in a positive energy status. We have to be high energy in order to create muscle. Because why would our body make muscle if we're not able to fuel the muscle that we already have? Right? It kind of starts making sense. Summary slide of all of those cell signaling principles, all those cell signaling molecules. This type of stuff will be on the test. I'm going to push you guys to learn it because it is super important. And these pathways are the way a lot of supplements try to target. This is the way a lot of exercise tries to target. This is the way drugs try to target. These are very important things for you to learn as you move forward as future clinicians, future exercise professionals, whatever. So now let's try to link this. Let's try to weave these two together. How does muscle physiology and systemic physiology really kind of come together? So our heart rate and our ventilation is going to be driven by our central and peripheral neural systems, right? So our ability to, the speed at which our heart is contracting is going to be under neural control, right? We have vagal tone or parasympathetic tone, and we have sympathetic tone. Our blood vessels are also going to be under control of the parasympathetic and sympathetic nervous systems. We can vasodilate or we can constrict. Remember, there's muscles that line the outside of the vasculature. So we're actually able to adjust what's going on with blood flow. Inside of our muscle, we have receptors that are going to respond to changes in tension or mechanical stimuli, temperature inside the muscle, as well as chemicals inside the muscle. So things like lactate or hydrogen ions are actually going to serve as signaling molecules to go to our brain. So as our muscle is contracting, it's actively talking to our brain. Our brain is then actively talking to our heart and to our body to control what's going on with blood flow, what's going on with oxygen delivery, what's going on with carbon dioxide removal. So when we're untrained, we have higher levels of the production of these signaling molecules, right? We have higher levels of productions of lactates, of hydrogen ions, whatever. As you get better trained, you produce the same amount of those, if not a little bit more, but your body gets better at handling the stimulus. So those stimuli are no longer novel to the body, and your body can say, "Hey, I've seen this situation before. I know how to respond to that. I've already adapted. I can respond to that a little bit easier. I can relax just a little bit in terms of heart rate. I can relax a little bit in terms of vomiting. I can relax a little bit right?" Because we've already seen it, we've already been there, we've already done it, we've already experienced it, we can actually respond slightly different. We actually improve our response to these stressors, to these negative things that would normally freak our body out. Because we've already seen it, we're already ready, we're prepared for it. So let's flip the script all the way around. What happens when you stop training? What happens with detraining? Well, basically flip everything, right? Like we have a reversibility here. We have a detraining thing that can occur. Essentially, everything is going to decrease. Our AVO2 max is going to decrease. Our VO2 max is going to decrease. Our stroke volume max is going to decrease. Our cardiac output max is going to decrease. The only thing that's actually going to increase is going to be our heart rate, because our heart contractility is going to decrease. So our ability to actually contract hard enough is going to decrease, and we're not going to be able to really match the performance of our heart. We're going to have to increase our heart rate in order to maintain that cardiac output. So we're going to have a complete detraining that occurs. Everything positive that we did is going to get undone when we detrain. The previous 40 slides have all been about aerobic adaptations. You can actually combine together periods of anaerobic exercise to get most of those endurance responses. So if we go through longer duration, short rest bout exercise, we can actually string together a lot of the adaptations. But when we start going through things like quick burst activity, like repeated 40s, repeated 50s, suicides, that type of stuff, that are short duration but high intensity, you're going to be more reliant on glycolysis and phosphocreatine system. So you're not going to get the same level of fat burning. You're not going to get the same level of transition from type 2 to type 1. But you're going to get better at that anaerobic sport. You're going to get better at muscle buffering. You're going to get better at handling acid loads. You're going to get better at hyperventilation and handling carbon dioxide. You're going to get increased hypertrophy, because what you're doing is you're essentially producing high levels of force rapidly and often. So you're actually going to get better muscle growth, whereas endurance activity, you're actually getting better muscle efficiency. You're also going to increase the ability to have PCR around, because you're increasing muscle volume, because you're increasing muscle growth. You're going to increase the amount of phosphocreatine that can be present. So you're actually going to improve that anaerobic glycolysis and true anaerobic energy system. There is some level of mitochondrial biogenesis that's going to occur with HIIT training. I'm not willing to sit here and say that the level of mitochondrial biogenesis is going to be as high as the aerobic, but you still are going to get aerobic adaptation when it comes to mitochondrial biogenesis. All right, lot of information for you guys. This is super important stuff. Again, like I said, this is what's some of this information. It seems awful, but this is what's going to help you prepare for being a physician, being a clinician, going into athletic training, physical therapy, health and fitness, whatever you're trying to go into. Drugs are going to be targeting these things. Everything is going to be playing through these central mechanisms when it comes to endurance training or exercise training or even recovery. So pay attention to it. I'm sorry that it's tough, but we'll get through it. Remember, I'm always here for you.